have discussed the basic things in the toxicology chapters. So many people have joined newly. I don't know whether they have attended the classes or not. So what do you mean by toxicology of insecticide? You people? What do you mean by toxicology of insecticide? Hello? Hello?
Yes.
What do you mean by toxicology of insecticide? Those who have not attended the earlier classes. Tell me anything about toxicology of insecticide. Anyway, we are going to start the new chapter, that is Unit 11, Synthetic Insect and Control Agents. What is meant by synthetic insect and control agents? Why do you understand by this title what is synthetic insect and control agents? Anybody? What is synthetic insect and control agents? What it tells us this chapter? What is the objectives of this chapter? Anybody? Synthetic insect and control agents. What is the objectives of the chapter?
No, sir.
No. Okay. Let us observe the objectives of the chapter is to study the chemicals that do not kill the insect directly, but it can reduce the population of the insects directly. So that's why we are using the synthetic insect control agents. So in the other earlier classes, we are using we have used the insecticides. So that insecticides directly killed the insects. So we have talked about the many type of methods to kill the insect in the earlier classes. But in the chapter, we are going to talk about the synthetic insect control.
Useful insects, sir.
What, Amma?
Useful insects, sir?
Not useful insect. This will control the insects, but not killing the insects. We can reduce the population of the insects.
Okay, fine.
Okay,
Have you
sir.
got it, my point?
Yes, sir.
You cannot kill the insect, but you can control the insect population by using various insects agents. That is the synthetic agents. Okay, we will talk what are the synthetic agents, then how to control the insect population. So that all we will talk in the last chapter, in the next chapter. So this is only the introductory part of the synthetic control of insect. So in the chapter, we cannot kill the insect directly. You can use some kind of chemicals to reduce their population, like affecting their behavior, then genetic material, then to reduce the growth of the particular insect. So for this purpose, you can use many kind of insect to reduce the population. So if you use increased insecticide, it result to several resistance problem. If you keep on increasing, if you keep on using these, insecticide agents, what will happen? That insects get the resistance problems. Means the insects get resistance to the particular insecticides. So that's why you cannot use overuse of insecticides. Okay, in the last chapter we talked about the what are insecticides and all those things. But in this, some of the insect chemostimulants are present. What are the chemostimulants? Insect chemostimulants. Insect chemostimulants are nothing but it is a nippling in early
Sterile.
50s. What happened, Amma? Nippling.
Nothing, sir, nothing.
You can see the insect chemostimulants. Insect chemostimulants are nippling the early 50s floated in the idea that reproductive potential of insect can be reduced by sterilizing the releasing of sterilized insect to compete with the exciting natural population. Meaning, insect chemostimulants are nothing but to reduce the fertility rate in the male insects. So that when it is fertilized with the female one, it cannot give the fertilized offsprings by using the insect chemostimulants. So that it cannot give the next offspring. If it is cannot give the next offspring, so population size of the particular species will be reduced by using a synthetic agent. One kind of synthetic agent is nothing but insect chemostimulants. Chemostimulants are nothing but it is reduced by the sterilizing, or it reduces the fertility capacity of the insects. So then the insects becomes a sterile. Sterile means it cannot give the next offspring. So in this you have one method which is called male sterile technique. Male sterile technique. What do you understand by sterile and fertile? Anybody?
Sterile means not producing offsprings.
Very good, very good. Fertile?
Produce
Can produce offspring.
Yeah, which can give the offsprings. But if you use the insect chemostimulants, insect chemostimulants is one kind of synthetic insect control agent which can be prepared by chemically. If you use this insect chemostimulants, the fertility activity of male insect will be reduced. If the activity of the male insect will be reduced, it cannot produce the offspring. So that a population of the insects will be reduced gradually. Not using the insecticides. We are using the synthetic insect control agents. Insecticides are different. Synthetic control agents are different. Those who have attended my past classes in toxicology, we have talked about many insecticides. By using the many insecticides, you can kill the insects directly in various crops. But here we are not going to kill the insect, but you are going to reduce the population of insect by using a synthetic insect control agent. One of the synthetic insect control agent is insect chemostimulants. If you use the insect chemostimulants, it will reduce the fertility rate of the insect male one, so that the insect becomes sterile. It cannot if it is made with the female also, it cannot give the next generation. So that gradually insect population will be reduced by using this synthetic agent, insect agent control. That is one of the method. We can call it as a male sterile technique. This male sterile technique is used for the eradication of screwworm. Screwworm is one kind of insect, and Coccilemia, it is a one kind of insect. So we have a many example for the insects. We can reduce the fertility rate so that the insect becomes sterile. It cannot give the next population. So this is the one kind of synthetic insect agent control to reduce the population rate. So there are many methods to reduce the population rate. We have a the following factors also used to important for access of male sterile technique. So male sterile techniques, we have a following methods. One of the following method is a method of mass rearing must be available so that a large population can be bred under laboratory conditions. And the next method, the method of sterilization should not adversely affect the mating behavior of the insect. Then we have the female of the targeted species should be preferably mate only once in their lifetime. So whatever the sterilized males are there, that sterilized males will be mated with the female one only once in a lifetime. If you keep on mate with the male one with the female, it gets the resistance power. So it may produce the offsprings due to getting of resistance of that particular synthetic control agent. So the population density of the insect must inherently be low or should be first brought down to low level. So these are the methods to reduce the fertility rate in the chemosterilant technique. Then you have some alkylating agents. So this is also one of the methods. This is called alkylating agent. This alkylating agents also reduce the population of insect. It is a one kind of synthetic insect control agent. So what you might have observed this alkyl group in the chemistry. So what is meant by alkyl? What is meant by alkyl group? So alkyl group is nothing but this alkylating agents act as an anti-metabolites. These anti-metabolites act by replacing the hydrogen atom of a molecule by an alkyl group. Methyl, ethyl, these are the alkyl group. So in this, hydrogen will be replaced. So this is also can be used by synthetic control agents. So structurally, they have no similarities, but all have the property of combining with electron-rich centers. So it is kind of electrophilic, which accept the electron pair and form the carbon in an organism. Alkylating agents radially react with the nucleophiles, which are polar compounds that donate an electron pair to carbon in organic molecules. This is one kind of a chemistry of what is alkylating agents. In this, alkalizing agents also useful for the reduce the population of insects. So which will act on the nucleophiles. The target of this alkylating agent is nucleophiles, especially insect gametes. So what is meant by gametes? Anybody?
Sperm cells and ovum.
Male and
Yes.
Reproductive cells.
Yeah. Ovum and sperm are the gametes. If you using this alkylating agent, the target would be nucleophiles and especially the insect gametes, which act upon the ovum of the female and sperm of the male. So this is an insect. This is act on the reproduction activity, which will reduce the reproductive power of the particularly in insect to controlling the next generations. Some sterilants, antiplastic agents. These are all what are the alkylating agents? So some derivatives of alkylating agents are azaridins. Azaridins is one kind of alkylating agent in which the ring nitrogen is attached to PO, PS or PN moiety have been found to be effective chemosterilants. So generally,
నువ్వు ఉన్నావు కదా మనం చూసుకోవట్లేదు నేను క్లాస్ కి వచ్చేసాను కదా నేను క్లాస్ కింద నువ్వు వచ్చిన్నావు కదా ఇంత మంట పెట్టేస్తే ఉంటుంది అది నువ్వు చూసుకోవాలి కదా నేను వెళ్ళిపోయినాను నువ్వు ఇక్కడే నాకు నువ్వు షాప్ కి వెళ్ళిన విషయం నాకు తెలుసా? నువ్వు షాప్ పోయినావన్న విషయం నాకు తెలుసా? నాకు చెప్పినావా?
Madam, mute your mobile. Please mute your mobile. So some alkylating agents, for example, azaridins. So this azar is also one kind of ring nitrogen. You'll have a some kind of here are the structures. Then offload also one kind of alkalizing agents which act on the reproductive activities like which act on the gametes and ova to reduce the fertility activity and reproductive activity of the insects. So some of the alkylating agents are azaridins, and another one is offload. Offload is offload we have in a 224466 exists. These are the chemical structure of the offload. So this can be used as an alkylating agents. You have an offload to structural formation. So generally this offload, it is soluble in water. So some chemicals soluble in water, some chemicals soluble in alcohol, some chemicals soluble in chloroform, depending upon the compound or depending upon that particular substance. High temperature and moisture cause the polymerization of offload. Offload is one kind of alkylating agent. We have a structure of the offload in your textbook. So this upload will be dissolved in water, some may be dissolved in alcohol, some will be dissolved in chloroform. Yes. So this upload can affect differently in different organisms. This upload is one kind of alkylating agent. If you see in the vertebrates, if high toxicity of upload in the vertebrates, which causes damage to bone marrow. We include vertebrates or invertebrates. Are you comes under vertebrates or invertebrates?
Vertebrates.
Yes, sir.
In vertebrates, this upload can be damaged to bone marrow and germinal tissues. What do you understand by germinal tissues? Anybody have any idea? This upload is a one kind of alkalizing agent. It has some kind of chemical structure which may dissolve in water, some may dissolve in alcohol, some may dissolve in chloroform. But if high toxicity of this upload, which damages the bone marrow in the vertebrate, and it also damages and it also affects in the germinal tissue. What is mean by germinal tissue? Where it is present? What are the germinal cells?
Egg layers.
Yeah. Reproductive cells, we can call it as germinal layers or germ cells or germinal tissues. Reproductive cells, which the cells which present in the reproductive organs like testes and ovaries, that we can call it as a germinal tissues. If high toxicity of this upload, which may damage the bone marrow of vertebrate and it may damage the germinal tissues of the vertebrate, and it also affects the both sexes of the insect. It could be female or it could be male. So this is, and at 50% in food upload, it prevented 100% hatches in house flies. So this may damage the house flies and some kind of insects by using a synthetic insect control agent. That is one kind of alkylating agent. In that alkalizing agent, some of the derivatives of alkalizing agents are azaridins, and another one is offload. This is about the offload. Then after offload, you have a TEPA. TEPA is one kind of alkylating agent. This TEPA is a phosphonic oxide. It is a colorless crystalline solid. It doesn't have any color. This TEPA will be in the form of crystalline solid, like a sugar. It is in a crystalline structure. So like that, with a melting point of about 41 degrees centigrade, this TEPA is nothing but one kind of alkylating agent, which doesn't have any color. Hence, this TEPA you can call it as a colorless. It is in the form of a crystalline solid, crystals form. It will be in crystals form. But it has some melting point. That melting point would be 41 degrees centigrade. Highly soluble in water. This TEPA is highly soluble in water and also soluble in organic solvents. But this TEPA is highly toxic to mammals. It causes a sterility in the houseflies at 0.1% in food. So, which makes the sterility of the housefly, meaning it cannot use food for fertility. So, if it causes a sterility in the housefly, the fertility rate will be decreased in the housefly. So, gradually the population of the housefly will be reduced. So, that when if TEPA is in a high toxic, it also sterilizes both sexes, not only in female insect, not only in male insect. If TEPA is high toxicity, it causes the sterility in the both sexes. It could be male or it could be female. So this is all about the TEPA. TEPA and afloat are the alkylating agent. Then you have a METEPA. METEPA is also one kind of a derivative of the alkylating agent. It is a phosphonic oxide. This color will be different. This is a METEPA is a straw colored liquid. This color will be straw colored liquid. This highly soluble in water and also it's soluble in solvent, organic solvents. This is a METEPA. METEPA is a one kind of alkylating agent. The color would be straw colored liquid. It is soluble in water and organic solvents like a afloat and TEPA. The same characters will be like a afloat and TEPA. It is also effect against the both sexes. This also acts in the both male and female sexes to reduce the reproductive activity of the insects. So that population size of the insects will be decreased. That is the kind of alkylating agent. You have a next non-alkylating chemosterilants. That is an alkylating. This is a non-alkylating chemosterilants. Non-alkylating chemosterilants. You have a major group, majorly 4 groups that are phosphoramides, melamines, and diobiterates, and diazoxylam salts, and miscellaneous compounds in general. Non-alkylating agents are more species specific than alkylating agents. Species specific. What do you mean by species specific?
It affects particular species.
Very good. Which affect the particular species. One compound is specific to one species and another compound is specific to another species. That has an property that property we can call it as a species specific. It's not like a alkylating agent. For example, phosphoramides. Phosphoramides replacing azidyl groups in alkylating agents with alkamino group yield a phosphoramide, insect sterilant. This is also one kind of insect sterilant. Example of this include hempa, and it is sulfur analog thiopempa. So let us see the hempa is a sterilized male houseflies. Sterilized male houseflies effectively. This hempa is one kind of chemical substances. It belongs to non-alkylating chemosterilants. This hempa is majorly effect in the male houseflies. This EMPA makes the houseflies to sterile. It cannot help to fertile. So that this is also reduce the reproductive activity of these houseflies, especially comparable to physiological effects to those alkylating agents. However, toxicological investigation shown in the possible carcinogen. So this is the structure of EMPA and thiohempa. This is the chemical structure of EMPA and thiohempa. After that, you have a melamines. Melamines also, it is a kind of alkylating agents. So this group are replaced by diethyl amino group. Some chemistry is involved in this substances. So you need to go through this, so you will understand. But the important activity of this EMPA and melamines and alkylating agent and non-alkylating chemosterilants are to make the insects to sterile. So if this chemical to make the insect to sterile, it cannot give the offspring for the next generation. So by using this, insect population can be reduced. The next you have a miscellaneous compounds. Miscellaneous compounds is nothing but a group of compounds. This, for example, folic acid antagonistic, then aminopterin inhibitors, then 5-fluoric acid, meiotic poisons. So this and some nitrogen mustard can be used to inhibit or to stop the activity of cell division of the nucleus in the insect. So that is also one kind of method to reduce the population of the insect. Then next you have a insect growth regulators. So insect growth regulators. So what do you mean by insect growth regulators? Do you have any idea about the insect growth regulators? Anybody?
Urea.
Anybody? If you have anybody have any idea about the insect growth regulators?
They inhibit the growth of the insects.
Insect growth regulators. What is meant by insect growth regulators?
Maybe hormones.
Maybe very good. Some hormones will help the growth of the insects. That is called some growth hormones. Okay. So what are the insect growth regulators? So like a human beings have an human beings have a growth hormones like a growth regulator activities like a hormones, the enzymes, different kind of physiological activity takes place in the human body. In likewise, the insect also some kind of growth regulators. What are those growth regulators? Are nothing but chitin synthesis. A chitin is an odd substances which are covered and exoskeleton of the insect. That is called a chitin synthesis. That chitin synthesis is present in the insect exoskeleton. Then these insects have some kind of hormones that is called a juvenile hormone mimic. Then you have another Equidisis. What do you understand by Equidisis? This Equidisis will be performed by many insects. Have you observed what do you mean by Equidisis?
Opposite to normal growth.
What am Equidisis is nothing but shedding of the outer layer of an insect, like a snake and other insect will shed their skin, outer skin. So that is called Equidisis. When it is shedding their outer skin, then new skin will be formed in the replace of the old skin, so that the insect will be increasing their size or growing their body size. But if you use the insect growth regulate inhibitors, so what will happen? If you inhibit the chitin synthesis, what will happen? Inhibit is nothing but to stop the chitin synthesis. What will happen, Amma, if you stop the chitin synthesis in insects?
It cannot be grow.
Yes, it can grow. And if you stop the inhibit of the juvenile hormone, will it increase their size?
No, it cannot increase its size.
Increase its body size. Yeah, it cannot increase its body size. And if you cannot perform the Equidisis process or shedding the skin of the particular insect, will it increase their body size?
No.
No. So this is also one kind of synthetic insect control, synthetic insect agent control. This is one kind of agent to inhibit the activity of chitin synthesis, to inhibit or to stop the juvenile hormone and Equidisis process. Let us see chitin synthesis inhibitors. What are the chitin synthesis inhibitors? Chitin synthesis inhibitor consists of various compounds acting on insects of different orders by inhibiting the chitin formation. Means if you are using the chitin synthesis inhibitors, these chitin synthesis inhibitors to helps to stop the synthesis of chitin formation in the insect. If it is stop the chitin formation of insects, so it cannot synthesize the chitin on the outer surface of the insect. So there will be no growth and size of the particular insect by using the chitin synthesis inhibitors. It is also a negative effect for the formation or a synthesis of chitin. If you use the chitin synthesis inhibitor, this is also one kind of a synthetic insect control agent. We are not to kill the insects, but you are stopping their growth, you are stopping their fertility rate, you are stopping their exoskeleton formation, you are stopping their chitin synthesis formation, so that it cannot become an adult, it cannot give the next generation. These are the some of the ways to control the insects. So one of the insect growth regulators, we have a benzophenyl ureas. This is also one kind of insect growth regulators. This is a Phillips Duffer company discovered the insecticidal activity of benzophenyl urea analogus. So in the first analog, which was shown in the effect to against the insect, was coded name. It resulted from the combination of herbicide diclobin with the urea herbicide. The chemistry and symptoms of the benzophenyl urea analogs were unique and not similar to insecticides. This benzophenyl is one kind of synthetic control agent which cannot kill the animals. So which cannot act
Excuse me, sir.
Please
share the screen if possible, sir.
Tell me.
Please share the screen if possible.
Yeah, next class I'm going to share the screens. So today I'm not prepared. Okay.
Sir, I have one doubt.
You can ask.
What is the use of these chemicals? Means they are stopping their sterility, but they will lose our crop, na, sir. They will all eat. Means crop yield will be less, na. So what is the use of these chemicals? No need now. We should completely kill these insects.
So some of the chemicals to kill the insecticides that are called insecticide, that chemical will kill the complete insect, right? So some of the synthetic insect control agents are to control the population of insect. You should not kill the insects anamata. మనము కొన్ని ఇన్సెక్టిసైడ్స్ ని యూజ్ చేసేమో కంప్లీట్ ఇన్సెక్ట్ ని కిల్ చేస్తాం. కానీ ఇక్కడ కొన్నిటిని ఏం చేస్తే ఇన్సెక్ట్లను చంపద్దు. చాలా ఇన్సెక్ట్స్ మొత్తం ఎరాడికేట్ అవుతున్నాయి, ఎక్స్టింక్ట్ అవుతున్నాయి. బట్ వాటి పాపులేషన్ ని మనం పెరగకుండా చూసుకోవాలి. సో అట్లా కంట్రోల్ చేయడానికి కొన్ని కెమికల్స్ ఉన్నాయి. సో ఆ కెమికల్స్ ని మనం ఇక్కడ డిస్కస్ చేస్తున్నాం. ఆల్కలేటింగ్ ఏజెంట్స్ అని.
Benzophenol ureas is a herbicide, no, sir?
హెర్బిసైడ్ అంటే దీంట్లో కాంబినేషన్ వేరే ఉంటది. బెంజోఫినాల్ అనేది ఒక కాంబినేషన్. మళ్ళీ హెర్బిసైడ్ కు వేరే కెమికల్స్ కూడా ఉంటాయి. ఆ హెర్బిసైడ్స్ మొత్తం కిల్ చేసేస్తాయి ఇన్సెక్ట్ ని. కానీ ఈ కాంబినేషన్ లో ఉన్నాయి మాత్రం వాటిని కంట్రోల్ చేస్తాయి. ఇట్ ఈస్ నాట్ లైక్ దట్ కైండ్ ఆఫ్ ఇన్సెక్టిసైడ్. ఇట్ ఈస్ ఎన్ ఏ కంట్రోలింగ్ ఏజెంట్. ఓకే.
Okay, sir.
Chemistry of benzophenol urease. Then the mode of action, how it is going to perform that particular benzophenol action, urease action in an insecticide. Chitin is one kind of polysaccharide. You know, chitin is one kind of polysaccharide. Chitin is a hard shell. This is present in the exoskeleton of the insect. It is a one kind of polysaccharide, so which made up of N-acetyl D-glucosamine units with some D-glucosamine linked with B1,4 glucoside bonds. And chitin biosynthesis from glucose takes place by the amination of phosphorylated sugar. Some chemistry will be involved in this mode of action because it is a chemical substances. So you more focus on this kind of chemistry. Acetylation results in the formation of acetyl glucosamine 6-phosphate. This amino sugar is activated by combining with UDP and resulting the UDP and acetyl glucosamine polymerase to form the chitin. Polymerization step is catalyzed by a chitin synthetase or chitin synthase. The enzyme chitinase breaks the chitin back to its monomers, that is N-acetyl glucosamine and glucosamine. So this is a one kind of chitin which is formed in the insect exoskeleton. So then you have its biochemical effects on benzophenol urea, insects that are related to the molting process, and since this physiological system which is primarily disturbed by these toxicants. So majorly this benzophenol ureas are majorly disturbed in the insects such as the target organs or molting process. Insects undergo molting process. What is meant by molting process?
Removing their chitin.
Removing their exoskeleton.
Chitin.
Chitin or remove their exoskeleton. So that process will be disturbed by using this benzophenol urease. So the insect's growth will be reduced, and which also disturb the physiological systems. All the physiological systems will be disturbed by using this benzophenol urease. And also this inhibit the DNA synthesis in the insect, which stop the DNA synthesis in the insect. And the inhibition of DNA synthesis may also relate to the cytostatic activity for fly imaginal discs. So apart from this, the effects of benzophenol urea treatment follows consistent pattern and indicate that the primary site of an action is disruption of chitin synthesis. In general, the insects die immediately at the time when chitin synthesis is critical or survival. So if it is high dose of this benzophenol urea treatment, so sometimes these insects will also die. The effects may be categorized as a disruption of equidisis. So benzophenol is disturb the activity of the equidisis, and which also disturb the failure to feed. If you use this benzophenol to the insects, which disturb the activity of equidisis and also disturb the activities of failure to feed and which also disturb the activities of delayed mortality and ovicidal effects. Let us see what is the disruption of equidisis. After treatment with benzophenylurea, symptoms are generally not observed until the molting process is initiated. Aploisis is completed normally following treatment. However, equidisis is disturbed. Severity after disruption of equidisis is generally dosage dependent. If doses will be high, if doses will be less, depends upon the dosage, the activity of equidisis is affected.
Sir, what is epolysis?
What, Amma?
What is epolysis?
It is a kind of treatment. Apolysis is a completed normally following treatment. Normal treatment, how much dose should be given to the particular insect to disturb their exoskeleton. So equidisis is prevented completely so that insect
protects the epidermis layers.
Huh?
Epilosis is to serve the protection against the epidermal cells.
What, Amma?
Epilosis
failure to feed. Then you have a failure to feed a number of insect species with some benzophenylurea analog and molt apparently normally. However, the mandibles and labrum may be displaced such that insect is unable to feed. So which disturb the activity of feeding mechanism also. That is called failure to feed. In this failure to feed, number of insect species with some benzophenylurea analogous molt apparently or normally. However, the mandibles, that is labrum may be displaced. Labrum is an upper lip, lower lips are present in the insect. So this labrum will be displaced such that insect is unable to feed by using this benzophenylurea. In other instance, backlash of the gut may lead to non-feeding. So the symptoms are generally lethal. Sometimes it's going to die. Lethal is nothing but it causes the death of the insects. But in some cases, insects may molt after an instar without feeding. So that is called stationary molt and regain the normal structure. So this is the effect of benzophenylurea in the insect to disturb the activity of feeding mechanism, that is called failure to feed. Then you have a delayed mortality and ovicidal effects. Treatment during the last larval instars can result in expression of symptoms being delayed until adult emergence. So the emergency of the insects will be delayed. Sometimes it affects the ovicidal effects. Ovicidal means which affect on the ovums. So the adult may not completely escape from the pupal molt, which results in the inhibition of flight and reproduction. In some cases, adult equidisis is completed, but the insect is morbiding and dies shortly thereafter. Ovicidal effects may also appear with the number of insects after treatment of adults. The embryos develops fully treated eggs, but completely developed larva fails to hatch. So larva cannot hatch due to the treatment of this benzophenyl. Direct treatment of egg is also effective in preventing hatching if carried out early enough after oviposition. Ovicidal effects have also been reported in instances when larvae consuming a toxic dose of benzophenylurea while chewing out of treated eggs. Such larvae usually are not able to establish feeding sites or molt to second instars. So this is an effect of ovicidal effects and delayed mortality effect by using benzophenyl activity. You have a diflubenzone. This is also one kind of synthetic insect control insect. Then you have a bupropigen. Bupropigen is one kind of synthetic chemical agent, insect agent. Some homopteran pests such as the greenhouse whitefly and Trilurodice and Vaporopyramine and sweet potato whitefly, so kind of insect will be affected by this bupropigen. Then you have one kind of juvenile hormone mimics. Juvenile hormone mimics. The juvenile hormone mimics are compounds bearing a structural resemblance to the juvenile hormones of insects. Juvenile hormones are lipophilic psychoterpenoids containing an exopide and methyl ester groups. Most of the JH analog synthesis at the early stages, such as a phenol derivatives and joaboin and metropin, hydroferin, and quinoprin, were not sufficiently stable and active under field condition. Two insecticidal mimics of juvenile hormones are methoprin, so which bears a close structural resemblance to juvenile hormones, and phenoxycrab, which possesses a phenoxybenzoyl group instead of a carbon chain with an aperoxide. Both compounds are soluble in organic solvents and have an extremely low toxic to the mammals. Both methoprin and phenoxycrab mimic the action of these juvenile hormones on the number of physiological processes such as a molting and reproduction. Exposure to these compounds at molting results in the production of insect containing a mixed larval or pupal or larval adult morphologies. The efficacy of this compound is the greatest when normal juvenile hormones titers are low. Namely, by using the methoprin and phenoxycrab, the activity of hormones of the insects will be disturbed, and also reproductive activity of insect will be disturbed, and normal physiological activity also disturbed by using the methoprin and phenoxycrab. This mostly affects the juvenile hormone. If this juvenile hormone is disturbed, the size of the insects and reproductive activity of insect and normal physiological activity of insect will be disturbed by using this methoprin and phenoxycrab. So that is all about the juvenile hormone mimics. Then you have attractants. Then we have Equidisis and agnostics. Then you have an attractants. Attractants, chemicals that attract the insect for food, egg laying, and shelters are called attractants. So the term, these chemicals which cause insect to make movements towards the source. So these attractants chemicals acting in a vapor phase, causing an insect to behave in a way which results is moving towards the source of the material or towards the zone of preferred concentration. By this, the sex hormones also attractants like a sugar and molasses have been used for the attractants in the poison baits. Attractants are nothing but is a one kind of pheromones which are present in the insects, like in human beings, you have one kind of hormones. So in the insect, you have a pheromones. That pheromones will be disturbed or in this particularly insect. These are attractants are highly specific, species specific, Therefore, these are ecologically acceptable. Hence, they are important tools in the IPM programs. IPM programs are nothing but this insect pest management programs. So, other than the fact they are esters, there are no similarities their chemical structures. Several environmental and physiological factors affect their effectiveness and attractants. So it will be effects on the environmental condition, and some physiological factors also affect the attractants. So this attractants is the one kind of synthetic insect agent control. Then we have some kind of sex pheromones. Sex pheromones is a chemical or a mixture of chemicals that is secreted to outside by an individual and trigger an immediate response of the sexual behavior. Sex pheromones which are present in the insects, or these are made up of a kind of mixtures of chemicals which are secreted outside by an individual and triggers an immediate response of the sexual behavior in the receiving individuals. Most insect sex pheromones are secreted by females only. Most sex pheromones are secreted by females and act as orders of stimuli for males. These females will attract the males by releasing some kind of odors. Odors are nothing but a kind of a smell by using these sex pheromones. So we have some insects like sex pheromones present in the Bombax mori, after which several sex modes have been chemically identified of this. So the largest number of compounds, more than 80, is known from more than 120 lepidopteran insects. The carbon number of these compounds ranges from 10 to 21, the smallest being a disnol isolated from peach of the fig bearer and Ansseria, Intelia, and other largest octadisli estate of the sugarcane, molasses. This all will be used. Sex pheromones have been identified, including Lepidoptera species, Coleoptera species, Diptera, and Orthoptera, and Homoptera, and Hymenoptera. So, in this all kind of class orders of insects, you can observe the sex pheromones. Sex pheromones are released from various types of organs located in the definite parts of the insect body. Types of organs and their locations have some taxonomic correlation in the, but their detailed morphology differs from the regarding to their family and even from species to species. General female sex pheromones glands of almost all moth species are located in the intersegmental membrane. So intersegmental membrane between the 8th and 9th abdominal segments, this sex pheromones will be secreted. Most of these glands are irreversible folds or sacs, dorso-ventrally located in the intersegmental membrane. Then you have a monitoring of pest densities. Traps may be used for estimating the population levels of a particular species in species area for the purpose of timing to control the measures. This is also monitoring of pest densities. Then mass trapping. In this technique, the large number of traps are used to trap the males so that possibilities of mating are reduced. Trap design, a type of pheromone dispenser, light up the trap above. So here the male one will be trapped so that the male cannot fertile with the cannot mate with the female, so it cannot give the next offsprings. It is called a mass trapping. The number and distance between the traps and important parameters which need to consideration. This is one kind of technique to trap the male ones so that it cannot mate with the female. So next generation will be reduced. So apart from this, you have some of the species and pheromones and its structure also present in the tabular form, like a pink bollworm. Then it has a pheromone gauze plure, and it has a chemical structure. So like that, you have a tobacco, then it has a pheromone called J9, tetradicinal, and it has a chemical structure. Then budworm, Eleonthis, like that, Indian meal, moth, Polidia, Intrapenectella, and it has some pheromone that is called JDE 912, tetradicinal acetate, and it has a chemical structure. Once you go through this, so you will understand this thing. Then you have an insect repellents. What do you mean by insect repellents? Insect repellents are the chemicals which cause the insect to make oriented movements away from the source of the chemicals. So Browner defined as the repellents are the chemicals acting in a vapor phase, preventing an insect from reaching the target to which it will otherwise be attached. The term repellent is used rather than a generalized way. Feeding repellents is used for chemicals which are known to prevent feeding by insects. Repellents have been used in many forms, directly by impregnating clothing to keep the insect away. That is, camphor crystals are commonly used in woolens to repel cloth moth, and keeping neem leaves in a rice and other storage grains to prevent damage by storage of pests. Application to body parts to repel insects, especially the mosquitoes. Example, DEET, N-diethyl toluamide is the most widely used repellent applied to the body for the protection of main against the mosquitoes. Then biting flies and fleas.
Odomas is the example.
Burning of evaporating the chemicals to repel insect from the vicinity. Various formulations of altrin like oil, mats or vaporizing liquids to repel mosquitoes. Prevention of feeding by insects. Prevention of feeding by insect in fields like a fentanyl acetate and an argontin compound as a feeding repellents against codling moth and oriental fruit moth of apples. So you might have know the some kind of repellents that will know to control the insect agents by using neem leaves and some kind of chemicals, camphor crystals. By that you can control the insects that are all called insect repellents. The most common use of chemicals or repellents has been repel blood-sucking insects and vectors of human disease. Prior to World War II, the following chemicals were used to repel the biting arthropods. So you have some insect repellents. Their acute toxicity in this tabular form. You have a repellent that is called dimethyl oxalate. It is an LD52 rat per body weight of kg. Then this is a species repelled. Then Rutledge 612 LD50. This is useful for mosquitoes, flies, and fleas. Then N-butyl acetaldehyde, this is a repellent we can use for the controlling of mosquitoes, flies, and fleas. Then you have an N-butyl acetylene, it is an LD50, so you can control the use for ticks and fleas. Then dibutyl phthalate is a repellent that can be used for only mites. These are some of the insect repellents and their acute toxicities. So this is all about the synthetic insect control agents. So you have some model examination questions, that is, write an essay on the chitin synthesis inhibitors. So already we have discussed about how this chitin will be synthesized in the insect, and how it will be inhibited by using different kind of chemicals. So we have discussed about the chitin synthesis inhibitors. So for this question, you need to write all the chitin synthesis inhibitors. That is the first question. And the next question, Yahweh, what do you understand by insect sex pheromones and how can this be used in the insect management programs? So for this question, we need to write the sex pheromones, all the story of the sex pheromones and how it is be used in the insect management programs, that is IMP. So you can write answer for this question about the sex pheromones, that is insect sex pheromones. Then write an essay on the use of insect attractants. So we have discussed in that attractants, that is insect attractants. You just write up the only insect attractants. It is a small question. It may be asked only for short way. You can need to write the attractants. Mostly, probably these three are the important questions from the chapters. One is the chitin synthesis inhibitor. Another one is the sex pheromones. The last one is the insect attractants. These may be probably come in the exam.
Sir, for chitin synthesis inhibitors, we have to write mode of action also, sir.
Yeah, mode of action also you can write. Because
it's
it is
long
asking a long question.
Yes, sir.
Yeah. So actually, this will be a dry lesson. There is nothing to explain. This only mixed with some chemistry. So once you go through it, you can understand what is what. Okay.
Okay, sir.
Anybody have any doubt? Shall I wind up? So in the next class, I will be shown as in the PPT form the next lesson. So probably you have completed the, I think 6 units or 50 units in this toxicology of insects. Only few students are attended in the offline. So I cannot repeat again all those. I'll go with the new ones. So once you go through these chapters, you will understand. Okay?
Sir, insecticides and cuticle is left, sir, third unit.
Adi explain. You just remind me. I will explain that. And also some postsynaptic membrane and presynaptic membrane also I left in the nervous system. So that also I will explain.
Okay, sir.
Thank you very much.
Thank you, sir.
Is your class over, sir?
Yes, over.
Okay, fine. Thank you, sir.
12 to 1:30,
Yes, sir. Thank you, sir.